I'm excited to be talking to you today about the draft FDA Bayesian guidance. I had the great fortune of being on detail from Vanderbilt University to FDA Center for Drug Evaluation and Research for 8 out of the last 10 years for part of my time, and I had the distinct privilege of working on various Bayesian working groups, FDA working with the very talented and committed statisticians in the Office of Biostatistics in Cedar who worked so hard to bring this draft guidance to us. This guidance is called Use of Bayesian Methodology in Clinical Trials of Drug and Biological Products Guidance for Industry. It was released last January 2026, and it's the longstanding efforts of many biostatisticians in the Center for Drugs and for Center of Biologics. There are immediate benefits to this guidance. It satisfies a long-time need at FDA. The Center for Devices and Radiologic Health published a Bayesian guidance 20 years ago under the amazing leadership of Bayesian biostatistician Greg Campbell. Drugs and biologics have never had a Bayesian guidance, and there was a feeling in industry that maybe FDA was not accepting of Bayes, but FDA never had any policy against using Bayesian methods in any certain context. The guidance legitimizes Bayes in general, not just for special cases, not just for boring information and rare diseases, not just for adaptive trials, but in general, and that's a very big deal. It provides a pathway for Bayesian designs and analyses, and it communicates to industry that Bayesian proposals are welcomed. It also increases the awareness of Bayes, and that will lead to an increase in exposure of people to Bayesian methodology in their training, both at pharma and other tech companies and FDA, and both statisticians and reviewers. So what problems did the guidance need to address? Well, as I mentioned, there was a reluctance of sponsors to use Bayesian methods in phase 3 clinical trials. No reluctance in phase 1 and phase 2, but in pivotal clinical trials, there was a reluctance. There was a need to allow clinical trial designs to be even more flexible than they currently are, to have better actionability of efficacy evidence measures. So, as we'll discuss later, the traditional approach is a bit indirect, and having more direct evidentiary measures leads to more clear and many times better decision making. Formal incorporation of extra study information is a well-known property of Bayes. It's very hard to do that without Bayes. Having a pathway to less arbitrary handling of multiplicities, I think, is going to be a byproduct of this guidance. Simplifying the paradigm. So what does simplifying the paradigm mean? Well, in the past, applications of Bayes were required to have a simulation or other calculation of the type 1 assertion probability, alpha. This is where a lot of the work went into designing clinical trials, because simulating alpha can be very complicated. The simulations have to take into account intentions to analyze, especially when there's adaptations, when there's multiple looks at the data, and that means that the simulations violate the likelihood principle. So anytime you have to factor in intentions, which is what happens in an alpha calculation, you are violating the likelihood principle. So this is an example of what goes wrong when you try to mix Bayesian and frequentist methods, and you ask a Bayesian method to satisfy a very non-Bayesian quantity of alpha. So this company, Asthmatics, had produced a product called Alair for refractory asthma. These are patients that would have to go to the emergency room, unable to breathe a few times a year. And so this was an invasive technique where you have a bronchoscope that's specially equipped to do ablation of tissues in the airways to make the airway bigger, so that during an asthma attack there would there would not be this loss of the ability to breathe. And so this was an amazingly well-designed study in which there was a placebo bronchial ablation. So this is extremely strong blinded control where the patients who were getting the placebo, they had the bronchoscope going down their airways to the same depth with the same rotations and the same sounds and the same length of time. So the patients couldn't tell if they're getting a real ablation or a placebo. And so most people would think that you're going to have a hard time recruiting patients for such an invasive procedure that's got a placebo in it. And so we'll see that that wasn't the case. So there were Bayesian decision criteria using a flat prior distribution, and the decision criterion was a posterior probability greater than 0.99 of a positive effect at an interim look, or a probability of benefit greater than 0.964 at the final look. This controls overall alpha at 0.05 if that were relevant to a Bayesian procedure. So the enrollment far exceeded the expectations. These were desperate patients. There was no time to do the interim look, and the final posterior probability was 0.960. So what does that mean? You failed to achieve your primary endpoint, which required a posterior probability of 0.964, and that was just calibrated or adjusted to satisfy the overall alpha with one interim look. So the primary endpoint failed because of an intended look that never happened. So a hybrid Bayesian frequentist procedure creates a lot of complexity and arbitrariness and confusion, and it violates the likelihood principle, which that principle means that data that could be obtained but were not obtained are irrelevant to your measuring evidence about an effect. So what about evidence quantification and actionability? There's been a very long-standing problem in statistics that most statisticians don't really want to talk about. Alpha was never an error probability, and that's at the heart of much of the issue. Alpha is the probability of making an assertion. So it's an assertion trigger probability. To be an error probability, an error means the same thing as a mistake. And so alpha would have to be the probability of making a mistake, and it's not that at all. And so you can't call something an error by assuming the very fact that would make it an error. So by calling alpha an error, we've endowed it with properties that it never had, and we've made it seem very important to engage in alpha spending instead of dealing with the chance that our decisions are correct, which is what Bayes deals with. So we were forced to translate a probability of getting data even more impressive than what we observed if the treatment doesn't work to an idea of the probability that the treatment works. So those probabilities have transposed conditionals, and to get from one to the other is a giant leap that makes traditional statistics actually extremely subjective. So this leap of going from a probability about data to a probability about the unknown efficacy is a very tricky and subjective leap. Regulators talk about regret, and a regulator's regret from the efficacy standpoint is approving a treatment that ends up not being efficacious. And so if you're interested in regulators' regret, that was something that alpha could never help you with at all. It's not something you can compute from alpha or from p-values. It's something that comes from Bayesian posterior probabilities. So if you have a posterior probability that efficacy is positive, given the data and given a prior distribution, the probability that the treatment doesn't work is just 1 minus the probability that it does work. And so that 1 minus probability is the probability of regulators' regret. And you can see from that formula that that has absolutely nothing to do with alpha. So asking a Bayesian to compute alpha is like telling a patient the specificity of a test, a medical test that he already underwent. the specificities of probability the test will be negative given there's no disease. Asking a Bayesian to compute alpha is like asking a poker player winning $10 million a year to justify his ranking by how often he places bets in games he didn't win, or by judging a politician by how often he speaks when he's honest instead of how often he lies when he speaks. You can see these have in common the idea of transposed conditionals. And so we need to be a little bit more impatient with transposed conditionals and work towards the probability of knowns given probability of unknowns given knowns. In other words, the probability that something will happen in the future given the past, or the probability that the drug is effective given the data. So Fisher had touted his approach as being objective, but he did that by really sneakily changing the question. The question was, does the treatment work, whether it's in agriculture or medicine? He changed that to a different question that could be answered without computers and without any prior knowledge. If the treatment doesn't work, how surprised should we be by data more impressive than ours? Notice the word more is emphasized. We're not calculating a probability of getting data as impressive as ours, because in the continuous case, that probability is 0. But we're calculating the probability that someone else's data is more impressive than ours if our null hypothesis is true for them. So a measure of surprise is not directly actionable to the things we're most interested in. So one way to summarize the difference in paradigms, which is quite a striking difference, is do you want the probability of a positive result, such as a large test statistic, when there is nothing, when the treatment is totally ignorable, it can't hurt you, it can't help you? Or do you want the probability that a positive result turns out to be nothing? So you have evidence that looks like a treatment is effective, and it turns out the treatment is not effective. So do you want the probability that a positive result turns out to be nothing? That is what Bayes is all about, and that's what we most often want. So what are the most important aspects of the guidance? Well, it documents that Bayes has its own operating characteristics. This may be the single most important part of the draft guidance document. These operating characteristics have absolutely nothing to do with alpha. They are the correctness of the decisions that you make from the data. Those are the Bayesian operating characteristics. So Bayesian operating characteristics can only be poor when the study generating prior is at odds with the analysis prior. So let's dig into what that means. So when you analyze the data, you'll have a collaborative project to arrive at a prior that's going to be used in analyzing the data. This prior in a phase 2 study might be using all kinds of information, and it might even be using some subjective beliefs used for internal decision making at a company, not for a pivotal phase 3 trial. For a pivotal phase 3 trial, unless you have amazing other data that's really provably relevant, your analysis prior is typically going to be serving the role of a restriction on what you know about the treatment. That restriction might be that we know the treatment is not curative, and so you can't have an odds ratio of 0. So whatever you're doing for the prior, the analysis prior, let's call that prior A. Now, when you're doing the simulation to show the Bayesian operating characteristics, you may say, well, everybody agrees with prior A to such a large extent that we're going to simulate our clinical trials under that same prior. And when you do that, you're going to show your Bayesian operating characteristics are just incredible. And that's just mathematics. But if you were to say, we want to do a bit of a sensitivity analysis, sensitivity to the choice of the prior, and we might envision a universe of randomized trials where the true effects have a distribution that's different from the distribution we're using as the analysis prior. So this prior S is called in the guidance document the sampling prior or the simulation prior, and I slightly prefer calling it the study generating prior, because that's the role it serves explicitly in your simulations. So you have a universe of treatment effects that's captured in this distribution S, and that is the distribution you draw from when you're generating clinical trials in your Bayesian operating characteristic simulation. So if the S prior is at odds with the analysis prior, your Bayesian decision will not be as great in accuracy as you think it is, but you'll find in many cases it's still excellent. And that's really what the simulations need to be showing. So we're going to simulate the probability of making a correct decision under a mismatch of the priors A and S. This link at the bottom is a presentation that goes into this idea in detail, showing you results of sort of in detail results of Bayesian operating characteristic simulations. So we are going to have in our simulation an example decision rule, and that rule is called E for we're going to decide to act as if the treatment is effective. And we're going to decide based on our rule here that if the probability that the treatment effect is working in the right direction, given the data and the analysis prior, is high, such as greater than 0.95, we're going to trigger our efficacy decision rule, capital E. So the question is, what is the correctness of that decision rule? So your primary operating characteristic is the probability that treatment is really effective. So when you say really effective, that's going to be, we don't know it's effective or not when we analyze the data. We have drawn a clinical trial at random. We've done the Bayesian analysis. Now, once the analysis is finished and you know that either E is yes or no, you're going to reveal the theta that generated the data. That's how you know whether your action is correct or not. So how does this play out? Let's say you're going to simulate 1,000 randomized clinical trials, and they have a universe of effects equal to little s. You're going to find the subset of those 1,000 simulated trials for which E is triggered under the analysis prior, A. The accuracy is the proportion of this subset for which the theta generating the data from S was actually positive. So let's say that of the 1,000 trials, you trigger a decision of effective in 200 times. Now you look at those 200 trials, what theta that was hidden to you a moment ago, let's reveal that theta. How often of those 200 times was theta actually positive? And so the proportion of the 200 times in which theta is truly positive is the accuracy of your positive decision in favor of efficacy. In a very similar way, you can quantify the accuracy of the other decisions. You might have a non-inferiority decision. You might have a similarity decision. You might have a formal inefficacy decision, which is related to futility. Another very important part of the guidance is it provides two distinct paths. The first path is when you don't want to think hard about the prior. And so not thinking hard about the prior often means to use a non-informative prior. That's going to make the Bayesian analysis more similar to a frequentist analysis if you have only one look at the data. But just remember what a non-informative prior is doing. It's saying that the treatment effect may be huge. I have a blog article showing an example where you're using a very, very wide prior, and that wide prior leads to a very bizarre assumption that the probability that an odds ratio is greater than 3 is higher than the probability that the odds ratio is less than 3. And I don't think many people would agree that that's good prior information or reasonable prior information. So you're allowing for a treatment effect of 10 to the 10th power. You're allowing for a hazard ratio of 0 when you have a non-informative prior. You're putting no restriction, and you're allowing very unstable results. And in the extreme case where you have 10 people on treatment and none of them had an event, you really don't want to tell the world that the probability that this treatment prevents that event is 100%. And so if you want to use a non-informative prior, so you're not going to think about the prior, then it may be reasonable to request that you demonstrate frequentist operating characteristics are okay. So you would actually simulate and go to these very troublesome simulations, especially if there's adaptation or sequential testing, and demonstrate that alpha is conserved. So that's path one. Path two is thinking hard about the prior. And this will be a lot of discussion, a lot of elicitation from people, collaborative effort between regulator and sponsor in many cases. And so if you really think hard about the prior and you justify your choice of the prior, and you also do the Bayesian operating characteristics where you allow a mismatch between the prior in the analysis and the study generating prior, but you still demonstrate that the efficacy decision rule is reliable, then you are demonstrating that you have excellent pure Bayesian operating characteristics. So it's not inappropriate to just keep emphasizing the primary Bayesian operating characteristic is the accuracy of the conclusions that you draw from the analysis. Now, multiplicity is not talked about very much in the guidance document, but what is talked about in the document is going to lead to better and better discussions about this. And I think multiplicity is something that all of us in our training have accepted it in a certain light without really thinking through the real scientific issues about multiplicity. So in a formal way, in the frequentist domain, multiplicity comes from the chances you give data to be extreme. And if you have multiple data looks, there's more chances for data to be extreme. That means your alpha goes higher than your nominal alpha. But multiplicity also comes from detaching clinical and statistical significance. A lot of multiplicity comes from using null hypotheses, the 0 point. And the reason I say that, and this is studied more in the likelihoodist school of inference, is that when you look at your times when p-value is less than 0.05, when you're simulating from a null case, a lot of those situations are where the estimated treatment effect is small. So it's, quote, statistically significant, but it's still a small treatment effect. And we also know even more clearly that if your sample size keeps getting bigger, you'll be able to declare statistical significance on a trivial treatment effect. And so you have two causes of multiplicity, and then we'll talk about the very general cause of just plain setting the bar too low. In the Bayesian framework, there's no multiplicity for multiple looks when you're talking about sequential looks. There's no mathematics that would allow you to do anything about it in the Bayesian context. There's no way to adjust what you're doing. New looks just make previous looks obsolete. So once you've looked after 100 patients and now you look after 150 patients, what you learned 50 patients ago is utterly forgotten and irrelevant to your look with 150 patients. So the idea of data becoming obsolete and the fact that Bayes is forward in time with all of its looks is what means there's no multiplicity. And there's no other real multiplicities in Bayes. All there is assertions of varying stringency. So if I told you that I'm either going to become an Olympic track star, or I'm going to become an NFL football player, or I'm going to win a neighborhood badminton game, you would say, there's a reasonable chance that you're going to be called a great athlete if that union of three conditions is what makes you successful. I have union things and made it much easier for me to be called a great athlete. That's just setting the bar very low, and it's just a fact of life. The probability of the union of a lot of events is going to keep getting bigger. So the challenge of multiplicity adjustments in the frequentist world it's not really seen as a logical thing to do outside of statistics. It doesn't really mesh with what people think the rules of evidence should be. So, to give one example, you're comparing A versus B. It might be treatments. And in the classical frequentist way of looking at it, you're going to discount your comparison of A versus B because you also examine C versus D. There's really no logic in that. And the way Bayes handles that is you'll have a degree of skepticism about the A-B difference going into the analysis, and you'll have a degree of skepticism about the C versus D analysis. Those two don't have any crosstalk. They don't really influence each other, but the skepticism you have for A versus B is what defines how you interpret A versus B, not the fact that you compared C versus D. So the multiplicity adjustments, besides having these evidence measures that don't really ring true to the rest of the world, is the adjustments are arbitrary because we don't have any general statistical principle that tells us, should it be Tukey's method, should it be Chaffee's, should it be Fisher's, Bonferroni inequality, multiplication inequality, false discovery rates. There's nothing in our general statistical principles that dictates those choices. They're fairly arbitrary. And the multiplicity adjustments are done on the randomness scale, and I'm going to argue they should be done always on the clinical scale instead of the randomness scale. And the clinical scale is the scale Bayes operates on. So Bayes gives you logical clinical ways to handle multiplicities. So we've miscast multiplicity as a randomness issue. It's something that you estimate things and then you get regular p-values and then you adjust them after the fact. So you're adjusting on a randomness scale, assuming like a family-wise global null hypothesis. But in a very general sense, multiplicity is setting the bar too low in the assertions you're making. So if you want to fix multiplicity, you need to not use an assertion that's a union of three athletic activities I might get involved in. You want to set the bar higher than that. So let's say you had 4 endpoints for a clinical trial and you wanted to calculate the probability of getting a treatment benefit on at least 1 out of 4 endpoints. Well, that probability is going to be pretty easy to be high. So that's a union assertion. What about an intersection assertion? Asserting that 4 things are simultaneously true doesn't need any multiplicity adjustment. That's a very high bar all on its own. So getting back to union assertions, make the bar high enough by demanding evidence for more than trivial effects. So you could say, we're going to call the trial a success if the probability of any mortality benefit is greater than 0.95, or you have strong evidence, 0.95 or higher, of at least a 10% reduction in a non-fatal endpoint. So you see, we've left this at the 0 point in our comparison because we say any here, but this one is demanding more than a trivial reduction in the rate of a non-fatal outcome. So that's one way to make things more stringent, raise the bar in changing the assertion. You might have, in the case of 4 endpoints, you might want to say, well, we need a high probability that any 2 of those are benefited by the treatment. That would be a different way of raising the bar. So let's change the subject just a little bit now and just look at where frequentists need to spend their time, where Bayesians need to spend their time. So frequentists spend a good amount of time, as they should, with data model specification. There's a few situations where you can be very non-parametric, but most of the time we need a data generating model, and it benefits us to have one. Where frequentists spend a lot of time is getting approximations to complex sampling distributions and using things like delta method, which don't even work that well. So the delta method works well when you have a sampling distribution that is almost normally distributed or at least symmetric. When the sampling distribution is not symmetric, the delta method will give you poor confidence coverage. The sampling distributions can get very complex, and in the case of sequential data looks in interim monitoring, the sampling distribution can easily be bimodal. or have a very bizarre shape, and most statisticians are not even able to derive those. There's other cases where we have approximations that are usually good, such as when you have a mixed effects model. But if you do a nonlinear mixed effects model, the approximations that go into that and that are relied upon for p-values and confidence limits are sometimes not good enough. And I don't think frequentist statisticians have been quite serious enough in validating the procedures. They're very quick to say to a Bayesian, I don't agree with your prior. And then the Bayesian says, well, that may change the posterior by 0.02 in this region, and your approximation for your p-value might be off by 0.03. Why is it you care about the prior and you don't care about your p-value calculation? Frequentists also need to spend time dealing with asymptotics, large sample theory, sufficient statistics, ancillarity, simulating alpha, which is very complex and must account for intentions, and then multiplicity adjustments get really dicey. Some of them are easy to apply, but you have lots of arguments about which one to apply. And engaging evidence for compound assertions takes a good deal of work in the frequentist paradigm. And there's other things frequentists have to spend time on. So where do Bayesians, well, before I get to where Bayesians need to spend their time, let's mention this. I already alluded to this. Frequentists don't spend enough time on sampling distributions. The whole idea of confidence limits and p-values is based on sampling distributions, but it's not always taken seriously. There's only a few simple cases where we get exact p-values and confidence intervals. And outside of a lot of those cases, we have non-normal situations and we have correlated data and whatever, and we're using a lot of approximations that are not always great. When interim analyses are done, the sampling distributions can be very bizarre. I have yet to see a single randomized clinical trial publication that included the correct point estimates and confidence limits when interim monitoring with potential early stopping were in play. It's very painful to do that. There are R packages that will do this for you, but I'm not seeing those used to give the correct sampling distributions. And you know that in the frequentist world, if you stop early for efficacy, your estimate at that time will be biased towards too much efficacy. Even commonly used models such as binary logistic regression and nonlinear mixed effects models have p-values and confidence limits of uncertain accuracy. Take, for example, binary logistic, where you have a very unbalanced outcome variable. The log likelihood becomes very non-quadratic, and our usual approximations are not always satisfactory. Where do Bayesians need to spend time? Well, they spend a lot of time with data model specification. This is where Bayesians and frequentists are in total agreement. They have to spend unique new time on the choice of the prior, and that's where a lot of the discussions and back and forth go into. They have to spend more time waiting for computation to be done, but if you have an array of processors, that becomes a non-issue. They have to deal with diagnostics. So whereas the frequentist statistician should be checking alpha and confidence interval coverage more often, the Bayesian counterpart to that is to make sure that you're actually sampling from the posterior distribution that you should be sampling from in your MCMC procedure. So there's a lot of diagnostics. These have matured greatly for checking the properties and convergence of your posterior samples of your parameters. In summary, the FDA draft Bayesian guidance establishes Bayesian methods as viable options in all contexts. It demonstrates the practicality of Bayes, establishes, very importantly, that Bayes has its own operating characteristics that are distinct from alpha. It justifies the use of pure Bayesian operating characteristics if the prior is well justified, or you run simulations that show that reasonable A versus S conflicts in those two priors still result in excellent decision accuracy. The guidance aids in understanding that alpha is not an error probability, never has been. It outlines approaches for prior specification. We'll need a lot more about that. It generates more exposure, interest, and demand for training in Bayesian methods. So there's some more information I have with various resources, my main navigational resources that address at the top. Much of the material I talked about was in a paper that Jack Lee and LaVange and David Spiegelhalter and I wrote recently in the Journal of the American Medical Association. So I'll refer you to that article. And then there's detailed simulations of Bayesian operating characteristics and explaining exactly what that means that I would refer you to. So I really appreciate everyone listening and hope you can go forth and keep increasing your interest in Bayesian methods.